Okay, good morning, and I hope that I haven't done something wrong. I don't see anybody in yet, but in any case, I'm going to start. Just to remind you all, please, that you have your second test on Friday, and exactly the same format as the first test. And the grades from that will appear as your midterm, although it's not really a midterm. It's just another test like the other three tests that you're doing during the semester, but that will go up as your midterm grade. Okay. I also remind you that tomorrow there is a review session, and the details for that are posted in Canvas. You will see that there are actually 2 review sessions posted, but the second one does not apply to you. It's the first one for tomorrow's review that applies. Also, we're not quite as far as that review may go. So don't worry about if they start talking about immunology, for example. Your test on Friday is only up to the end of today's material. I do not include what we'll do on Wednesday, okay? But that's a bit unfair. I only leave you a day to do it. So the test on Friday, same format as before, Canvas during the class time, please, and it will be multiple choice as always. Okay, so let's proceed with. Just let me check that I'm doing everything correctly. I get a bit worried when I see there's nobody in. Okay, no, I seem to be. Okay, all right. So this is where we finished off last time. Today, I'm going to finish off our nutrition and digestion, and then we're going to move on to gas exchange and circulation. And so this is where we finished off last time on the absorption of fats. Just very briefly, I'll repeat for you. Fats are, first of all, emulsified. That means they are broken into very, very small pieces. Fats won't dissolve in an aqueous environment, but they can be broken down into such small pieces, such small droplets, that they are easily attacked by enzymes. And the enzymes which break down the fats and lipids are the lipases. And lipases are secreted largely by the pancreas, some by the liver, some by the cells of the intestine itself. When you break down the fats and lipids, they get broken down to their component fatty acids, which are relatively small molecules, which easily pass across the cell membrane, because the cell membrane, of course, is lipid. So they dissolve across the cell membrane quite easily. The fats are broken down into fatty acids and what are called monoglycerides. That's a glycerol molecule with one fatty acid attached to it, which can also pass into the intestinal cells themselves. It's rather strange because they get broken down first, then they get rebuilt inside the cells. They're broken down so that they can pass across easily. They rebuilt into fats inside the cell. They're rebuilt into triglycerides. And those triglycerides are packaged like this. This is a phospholipid vesicle here. It's just got a single layer of phospholipids, not a double like a membrane. It's a single layer like that. And packed inside here, you can see all of these are triglyceride molecules and cholesterol all packed together. Then this phospholipid shell with proteins in it. And this is called a chylomicron. Very, very, very small. They're literally microns across. And these are excreted by the cell by exocytosis. And they are then picked up by the lacteal, which is that little finger-like projection of the lymphatic system inside the villus, inside the intestinal villi. And they pass into the lacteal, into the lymphatic system of the body, and they are then conducted all the way up and emptied into the blood circulatory system at the subclavian vein, which we'll hear about in a little while. But a rather complicated system for taking up, processing, and distributing the fats. And the fats then go off in the blood circulatory system to various organs which will process them, especially the liver. Okay, so in the colon, which is the large intestine, that has very little nutrient absorption ability, but its main function is to absorb water and some salts as well. And that concentrates the remaining material. But inside the colon, there is a huge, huge population of bacteria, which are very, very beneficial. And in us, for example, those bacteria contribute all sorts of vitamins, nutrients, things like that. But we're only just beginning to find out that it also has huge impact on all sorts of other physiological functions. And amongst them, by the way, appears to be neurological function. We don't understand all of the linkages, but it is becoming very clear that that large population of bacteria in the colon is extremely important to us. It's not an accident that it's there. It's a fully mutualistic relationship between us and our bacteria. Now, this does not apply to us, but in many organisms which depend more heavily on plant material, there's a part of the large intestine called the cecum, which is the first part of the colon. And the cecum, very often in herbivores, is important in breaking down cellulose. And for that, it has its own special population of bacteria and ciliates and things as well, which will break down cellulose. And in some herbivores, the cecum is very, very large and may be very elaborate in order to digest the plant material more efficiently. And an example of that in the rabbits, for things like that, for example, the cecum is very large. But in the human, the cecum does not perform that function. We don't have any ability to break down the cellulose, or at least minimal ability to break down cellulose. There may be some bacteria which are able to do it. That wouldn't surprise me. But we don't have any dedicated function of our intestine to breaking down cellulose. In the cecum of human beings, however, we do have, here's the cecum here. This is human. Here's the small intestine here. There's a sphincter here, which leads into this part. Here is the ascending colon, the transverse colon, the descending colon, and then the rectum. And where the small intestine joins, this is the cecum here. And we have an extension of the cecum, which is called the appendix. Now, this in itself is interesting, because the appendix used to be thought to be just a redundant kind of little organ. In things like the rabbits, the appendix is very large and elaborate, and it participates in breaking down of cellulose. It's part of the cecum, after all. But in humans, it's this little tail-looking thing, but it has a lumen. It has a space down the middle. And it used to be thought that in humans, this was just a useless little redundant part of the gut. Certainly, if we have it removed, which many of us do, it appears to have virtually no effect on us whatsoever. But it turns out now that, in fact, the appendix is part of the immune system. And it is part of the gut's immune system, and it has an important role to play in the maintenance of the immunity of the gut. We can do without it. Not great. Its functions can be taken over by other parts of the immune system if it's removed, but that in humans, that's its main function. It's part of the immune system of the gut. Once everything's passed through the colon, especially in the last part of the colon, as much water is extracted as possible. And the material now condenses into what we would recognize as being feces. And the feces are a mixture. There are undigested material, the products of the remains of digested material, a tremendous amount of fiber, at least it should be. We should, even though we don't digest cellulose, we should still eat a high amount of fiber to maintain bulk in the intestine, and that is what stimulates peristalsis in the gut. The stretching of the gut with fiber leads to increased peristalsis and efficient peristalsis. And then mixed in with that, with all of this waste material, is a very large amount of bacterial material. The bacteria in the colon especially reproduce very, very rapidly. And their population is controlled by a substantial amount of that population continually being voided from the gut. And those are the feces, temporarily stored in the rectum until they're eliminated through the anus. And the anus, of course, is a sphincter system, but a very, very intricate sphincter system. It is not a simple band of muscle. It's a very complex set of two different sphincters, which control bowel movement. I think that there is sorry. So that's the last that I want to say about so far about the actual mechanisms of the gut. There is a slide, a very last slide on this set. It's a kind of a summary slide. It's just a diagrammatic version of the interaction of the gut with its accessory glands and then with the circulatory system. Make sure that you understand all the different parts. And as far as possible, remember what enzymes contribute at each stage. I do want to spend just a minute talking about homeostasis in the body. Now, this is discussed in detail in that section, Animal Form and Function, which I asked you to do on your own. But this is such an important section, and it applies very strongly to what we're talking about here in animal nutrition. I thought I would just cover it briefly. Many of our physiological functions are very carefully regulated with mechanisms to maintain homeostasis. Homeostasis is optimal functioning, to keep our physiological systems within an optimal range of functioning. And this is most usually done by feedback mechanisms. Just 1 minute, sorry. Is most often done by a set of feedback mechanisms. The body continually monitors its status, and by monitoring its status, able to modulate its responses so that it keeps the body within optimal bounds. And that is what we refer to as homeostasis, and it is maintained by homeostatic mechanisms. Here's a simple example, and that is the maintenance of body temperature. So our internal body temperature is very carefully monitored, and is monitored by a part of the brain called the hypothalamus. The hypothalamus actually has blood passing through it, obviously, and the hypothalamus monitors the temperature of the blood passing through it. And the optimal, it varies from person to person, by the way. This is not if you take your temperature and it's a little bit, your normal temperature is a little bit different to what you read being normal temperature, don't worry about it. There is a variability. But in general, body temperature is between 36 and 38 degrees Celsius. And that is for human beings, the optimal. 36 to 38, and you will often see 37, which is in between the two, obviously. And that is your internal body temperature. It's the temperature of blood coursing around your body, bathing your organs, and they are maintained at that optimal temperature. And all of our enzymes, for example, operate optimally at about 37 degrees Celsius. Take it above and take it below that, and our enzymes simply don't operate very well. Take it too far away, and our enzymes will actually stop functioning. So it's very important to keep it within that narrow bound. But our temperature does go up and it does go down. It goes up and down in response to all sorts of things. It'll go up in response to exercise, for example. Our muscles generate a tremendous amount of metabolic heat. So you can go up if you exercise. Go outside in the very cold weather and or in cold water, and your body temperature may begin to drop and the internal temperature may begin to drop. So let's see what happens if the temperature drops. The stimulus will be that decrease in blood temperature, and that is picked up by the hypothalamus very quickly. It very quickly receives that and will respond. So there is a sensor, and that is the hypothalamus. And it's going to send messages. As soon as the body temperature starts to drop, it will send messages. It doesn't show it here, but what it sends messages to especially is to the thyroid, because the thyroid modulates our metabolism. And it will send a message to the thyroid, and the thyroid will send out hormones which will stimulate the metabolism. And there are three things basically that happens. First of all, our overall metabolism is raised. So we start to generate more body heat. The second thing that happens is that it will send instructions to especially to your peripheral blood vessels, and they will contract. And the reason for that is that they begin to direct blood to the core of the body. So, and they stop being cooled on the outside, but they also redirect all the blood to the core and especially to the brain. The third thing that happens under the influence of thyroid hormones is that we begin involuntary muscle action. We begin to shiver. And our muscles start doing this. This is familiar to all of us when you're very cold and you start shivering. What is happening is your muscles are contracting, relaxing very quickly, and that generates body heat. And as a result of all of these things, the body temperature begins to rise. It will actually start to rise quite fast. And we now run the risk we can begin to overheat. If we start to overheat, again, the hypothalamus is still monitoring the blood temperature. Now, the hypothalamus picks up that we are starting to heat up, and it will send out messages to the body to counteract this and bring the temperature down. There are two major ways in which this is done. There is, first of all, that thyroid stimulation is stopped completely so that the metabolism does not increase at all. But very importantly, we begin to sweat. Sweating is one of our major cooling mechanisms, and it works because water has such a high latent heat. It takes a lot of energy, a lot of caloric energy to get water to actually turn from liquid to gas. So when we do sweat and the sweat evaporates, it takes with it a tremendous amount of heat. This is a very efficient cooling mechanism. And the second thing is that it gets our blood vessels to dilate. So, for example, in the palms of your hands, the soles of your feet, except that doesn't work very well if you're wearing socks and shoes, but the palms of your hands and very importantly, your face. The blood vessels dilate and they start to radiate heat. And that also a major heat loss. As a result of all of these, the body temperature decreases. What if the body temperature decreases too much? Boing, you go into this circuit. What if it increases too much? Boing, you go into this circuit here. And the net result is that we modulate between, let's say there's wild swings, the swings will start to even out until you end up at an optimum temperature here. So that is a major homeostatic mechanism, and we say this is by feedback. This here, when we increase the body temperature, we cause an effect. If we cause it with this circuit here, we cause an effect. The effect is to raise the body temperature. The body picks that up, picks up what has happened. It's a feedback, and it begins to pull. It begins to act against that, to pull it into a homeostatic range. And this is a feedback. There are a few feed-forward mechanisms. A feed-forward mechanism is one where when we produce an effect, it amplifies the effect. This here, we produce an effect, we immediately start to modulate that effect, bring it down. Feed forward, an effect increases the effect. And I'll give you two examples. The one is in breastfeeding. If a mother begins to breastfeed, as the infant feeds, it increases the hormones which facilitate production and release of milk. And as that increase and production of milk occurs, that acts as a feed forward. It increases the hormones which facilitate production and release of milk. And that in its turn increases, and this is a feed forward, so it amplifies. The other one we'll hear about when we talk about blood function and circulation, and that is in blood clotting. All that you need for blood clotting to begin is for the very, very start of it is a feed forward mechanism. The start of blood clotting increases the ability of the body to form a clot. Form a bigger clot, you increase the ability to form a clot even more. Those are feed forward. This is feedback, because we produce an effect, and that produces a counter effect. Produce an effect, counter effect, and between them they end up balancing one another out. So this becomes extremely important in our maintenance of energy stores in the body. We store a lot of our energy because in general, especially in our at our current status of human civilization, we tend to have a very rich sources of nutrient. We eat far more than we actually need. And the body stores energy-rich molecules in one form or another. Our very first store, our most immediately available store, is in the muscles and in the liver. And it's in the form of glycogen, this polymer, glycogen. All that glycogen is polymers of glucose, uses glucose as the monomer, joins them all together to form this complex molecule, glycogen. And this is our most immediately available store of energy. It's what we access just routinely. But we can also store energy in fat. And that is, it's very efficient, especially in terms of the amount of space. It takes up a huge amount of energy for a small amount of volume. But the fat is not that easily accessible. It's only accessed when all of the glycogen stores, for example, have been exhausted. In any case, what we have to have a continual circulating amount of readily available energy in the form of glucose. And our very first resort is to access glycogen. So, and that we'll just have a look at how that is done. Okay, let's first of all realize there is a certain level of glucose in the blood, which is optimal. And that's between 70 and 110 milligrams per 100 milliliters of glucose. And if we fall below that, we run the risk of starving cells of energy. But rising much above that is also extremely dangerous. The glucose becomes toxic to many cells if you raise the glucose too high. So the main way in which we modulate our glucose levels is by an interaction of that organ called the pancreas and of the liver. The liver, especially because it is the major store of glycogen. But this also does apply to the muscles, but we'll consider mainly the liver. So let's say that we have a big meal and we take in far more food, more nutrient than we actually require. And our blood glucose levels begin to rise, and they rise above the optimal levels. As soon as that happens, in fact, as soon as we start eating, the pancreas, which is an endocrine gland as well as a digestive accessory gland, the pancreas begins to secrete a hormone called insulin, which I'm sure you've heard of. Insulin percolates throughout the body. And what it does is the major effects are, first of all, that it increases the permeability of cells to glucose. Cells begin to be able to take up glucose. And this is especially the case in the liver. The liver can take up a huge amount of glucose. It also applies to the muscles. So we begin to actively transport glucose into the cells under the influence of insulin. The second thing is that the insulin stimulates the liver and muscles to begin to manufacture glycogen. So they turn the glucose into the storage product of glycogen. And the net effect of all of this is to suck glucose out of the blood, out of the serum, and the blood glucose levels begin to drop. But now, what happens if the blood glucose levels drop too low? What if they drop below 70? Well, the pancreas is again the sensor and produces a response. And under these circumstances, it secretes another hormone. When the blood glucose levels fall too low, it secretes a hormone called glucagon. And glucagon percolates to the liver and to muscles, and it causes the breakdown of glycogen and the release of glucose into the bloodstream. So what happens? The glucose levels begin to rise. But what if they rise too high? Well, if they rise too high, then the pancreas begins to secrete insulin. And the liver, muscles, everything else take up the glucose and make glycogen. We bounce between these two. We modulate by feedback until we reach an optimal, no matter whether you eat or whether you're starving. You eat your meal, but you have long periods in between the meals when you still have to maintain this. It doesn't matter. Under normal circumstances, the pancreas maintains, by feedback mechanism, maintains the blood glucose in this region here, 70 to 110. You will know that sometimes this does go wrong. Sometimes, for instance, the pancreas cannot secrete insulin or does not respond to the presence of blood glucose. And these produce the disease of diabetes. So diabetes is a malfunction of this homeostatic mechanism. Okay, now here, this is just, I'm not going to go over it. This is for your own summary, and make sure that you understand what secretions, what the major enzymes are that are involved, major secretions and enzymes are involved, what the function is, and how some of these interactions occur. Okay. All right. So now just give me 1 minute, please, because I thought that I had. I don't. All right. 1 second. Excuse me while I do that. Share screen. Oh, for goodness sake. Yeah. There you go. All right. Sorry. Okay. So I hope that you can all see that. If not, we're in real trouble. So we're going to move on now. Okay, we've talked about nutrition and everything. So what? I told you that everything absorbed from the gut goes to the liver, and then from the liver to the heart, right? Fine. All of that fat and everything, it went into the lymphatic system. The lymphatic system ended up emptying into the subclavian veins, which you'll see in a minute, so what? What is happening? Everything has to be circulated. All of the stuff that we absorb from our gut, all the nutrients, etc., have to be circulated. In addition, we are aerobic organisms. We need continual source of oxygen. We need to be able to expel our carbon dioxide, absorb oxygen, and circulate that. We have to get it to everything. So we're going to start talking now about how that circulation system actually works, because it's actually quite complex. In fact, we'll learn in this section, and when we talk about immunology, that there are two circulatory systems which run in parallel with one another. But we'll talk mainly this time about the blood circulatory system and about how gas exchange takes place, especially. So we're engaged in a continual exchange of materials with our environment, whether we're eating, digesting, absorbing nutrients, or whether we're breathing in oxygen and breathing out carbon dioxide. We are exchanging molecules with our environment. There are organs of exchange, but the molecules that are absorbed have to be distributed throughout the body to all of the organs. Every single cell needs oxygen, or even though the lungs absorb the oxygen we absorb needs to be distributed to every single cell in the body. In general, we exchange molecules we exchange substances with cells in the form of very small molecules, which are able to move by diffusion across cell membranes. And for the most part, we have to have mechanisms of taking those molecules and placing them close by to the cell membranes so that diffusion can occur. It's easy if you're a unicellular organism, right? Because you have just a single cell with a big surface area in relationship to your volume and stuff. Everything you need can just go across and straight into the cytoplasm. Much more difficult for a multicellular organism, where cells may be quite isolated. For example, cells of your toe are nowhere near your lungs, and yet they need the oxygen just as much as the cells of your lungs do. So multicellular organisms especially need some way, usually, unless they are very small, they need some way of circulating stuff around. And in general, as we become more complex, as the organisms become more complex, what they have is a circulatory fluid that is easily moved. They have a series, somehow or other, they have to pump that around the body. They have a set of interconnecting vessels, and they have to actually actively move that fluid. So they usually have a heart, a muscular pump, which will move that fluid. And when we look at different organisms, we see that there are many different kind of permutations of this basic structure. Circulatory systems, there we see recognized two systems really. These are either open systems, that is where there is a reservoir of fluid which percolate, which bathes all of the organs, for example, and that fluid is picked up by a vessel system, pumped around the vessel system by a heart, and then dumped back into that open reservoir. That reservoir is very often filled with a fluid which we refer to as hemolymph. It's not really blood, it's called hemolymph. In a closed system, instead, the blood is pumped around by pipes which interconnect with one another, so that the blood never actually leaves the vessels. But there has to be some way still of allowing molecules to leave the blood vessels and percolate into the tissues. So those open systems are very typical of things like the insects, mollusks, not all mollusks, but many mollusks. And then the closed systems are more typical of things like annelids, cephalopods, and then all of the vertebrates, which all have closed systems. So here's what we mean by an open system. This is typical for an insect. They have many vessels, which throughout the body, but the vessels are open, and they dump fluid into the body cavity, where it percolates around as hemolymph. And that hemolymph is then picked up very often by the heart itself. The heart often has big ports in it, which suck in the hemolymph and then pump it back around. This will usually percolate through some sort of respiratory mechanism. Here, there'll be some sort of respiratory mechanism which will oxygenate the hemolymph before it's pumped back out into the body. A closed circulatory system is one like this. There is a heart which pumps blood around in vessels, and then those vessels subdivide very small. Very often somewhere in here, there is a respiratory system which oxygenates the blood, and the blood is then pumped all the way. These small branches would go through all of the different organs and carry the blood and nutrients and oxygen, etc. Carry that to the organs. It has to diffuse across somehow, diffuse across the walls. We'll see later how this is done. So this is a typical closed system, and this would be typical of things like annelids and the very efficient, fast-moving mollusks like cephalopods, and then the vertebrates as well. So in the vertebrates, there is certain commonality in the vertebrates, and that is, first of all, all vertebrates have a closed circulatory system, and they have a distinct heart which pumps the blood around the circulatory system. We refer to it as the cardiovascular system. Vascular simply meaning the vessels, and then the cardio part is the heart itself. In the vertebrates, there are three main types of vessels. And the first are the arteries. Arteries always carry blood from the heart, and they carry blood under pressure of the heartbeat. Then the other kind of vessels are the veins, and veins have very low blood pressure in them, a very, very low blood pressure. And they carry blood which is deoxygenated. Usually, there's an exception to that, but they carry blood towards the heart. it is usually deoxygenated. Joining the two, joining the artery, because remember this is a closed system, joining the arteries and the veins are a set of very, very fine blood vessels called the capillaries. And we'll find the capillary beds all over the body. There are many, many places where arteries subdivide into smaller and smaller and smaller units. They branch into very fine units called the capillaries, which join to veins. So blood flow is one way in this system. It is one way. But there are complications, not complications, elaborations on this system. One thing I want to emphasize, the arteries and the veins are distinguished by the direction of blood flow, not by whether or not the blood is oxygenated. Arteries carry blood away from the heart. Veins carry blood towards the heart. That is their definition. Now, in the vertebrates, there are various kinds of heart and various kinds of circulatory systems as well, as we'll see. But in all of them, blood enters the heart through a chamber called the atrium and is pumped out by a ventricle. So it enters into a muscular chamber called the atrium. It leaves by a muscular chamber called the ventricle, but the ventricle is usually much more powerfully muscular because it's got to pump the blood. It has to pump the blood around the body. The branching of the arteries is complex. They divide first into little branches called the arterioles, which still maintain some of the characteristics of the artery, as I'll show you in a minute. And they then become capillaries and join up with the veins through the capillaries. Here, for example, let's just have a look. This is a simple vertebrate circulatory system, and this is in the fish. And have a look here, and you'll see that first of all, they have a system of circulation which takes the blood through the gills for oxygenation. So here's an artery leading from the heart to the gills. It's taking deoxygenated blood. Notice an artery carrying deoxygenated blood. That's important. To the gills where it is oxygenated. And it flows back through the body and it flows up into the heart. Here's the atrium here, and here's the ventricle. This heart is a simple elaboration on a tube. That's all. You've got one atrium and you've got one ventricle. And the heart pumps the blood in one direction through this, what is essentially a single circulation. It's a single circulatory system. Now, I'll just mention to you, because we talked about the evolution of the vertebrates, I will just mention to you that the lungfish have a separate circulation here. Not terribly efficient, but it goes through the lung. There are fishes with a lung, right? We know that. And that they have a separate circulation system. And the lungfish that can be taken out, that can live out of water, when they come out of water, they can shut off the circulation here, and move it through the lungs. Now that is very, very interesting because, first of all, in our system, in the higher vertebrates, there's an entirely separate circulation. They have lost the circulation through the gills in the higher vertebrates, and instead there's a circulation entirely through the lungs. So we're going to see this single circulation system can only work if the blood is going in just through one circuit, because we've only got a simple heart here that pumps in through one circuit in one direction. But as we go, if we look at the different kinds of vertebrates, we find that there are actually different kinds of heart. Here's the fish heart, a simple elaboration on the tube. But here is the amphibian. And an amphibian actually has a dual circulation system. The amphibians have taken that lungfish model and they have elaborated on it, as evolution always does. And they have produced a double circulation. It's not nearly as elaborate as ours, but it's still a double circulation. And in order to maintain that double circulation, their heart has changed. Here is the heart, but it now has two atria, one there and one there. And it has just one ventricle there. We'll see the function of this in a minute. I just want you to have a look at these hearts and realize that we are seeing, as we look at these, we're seeing elaborations of structure. Here's a reptile. A reptile really also is very similar in some ways to the amphibian, in that they have two atria here, and they have two ventricles, but the ventricles are connected. They are not completely separate from one another. And in fact, look at the amphibian and the reptile, we'll realize that blood can actually mix in the heart. It can mix somewhat between the atria, and it can mix quite a lot here. This is not a very, very efficient system of circulation. It works perfectly well. Even we know that there have been some enormous reptiles. The reptiles have developed somewhat of a separation of the two ventricles, but it's not complete. Same with the auricles, the atria rather. They are somewhat separate, but not entirely. But these systems work fine if you are not endothermic, if you are not maintaining a constant body temperature, or if you're not able to maintain a constant body temperature, then these systems appear to be sufficiently efficient to maintain that. But once you become endothermic, you need a very efficient system of aerating the blood and then circulating it rapidly and efficiently to the rest of the body. And that is what we see in the birds and in the mammals. In the birds and mammals, if we look at them now, we see the chambers are completely separate. There is a septum all the way through here. Each atrium is separate from the other. Each ventricle is completely separate from the other. We have the same system in the birds, and we have the same system in the mammals. And we're going to see now how that affects the circulation. Just keep this in mind when we talk now about the amphibia. And here we see, there's that heart, the chambers not completely separate from one another, but the blood by and large remains separate. The oxygenated and the deoxygenated blood are kept more or less separate from one another simply by the mechanics of fluid flow within the heart, but they do mix. There is a certain amount of mixture. But here we see it there. You remember it's got one ventricle and two atria. Here's the ventricle here pumping, and it pumps blood first to the amphibia have lungs. They can breathe. Can breathe fully terrestrially, but they also have another very important source of oxygenation, and that is that they have a moist skin, and they have abundant capillaries that run through the skin to aerate, oxygenate the blood and get rid of carbon dioxide. So this is called the Palmocutaneous Circuit. Here it is here. And this circulates blood both through the skin and through the lungs, oxygenates it, returns it to the heart, where it is pumped out to the rest of the body. So what we see here is that there is a double circulation. There's a oxygenation, decarbonization circuit there. And then there is a circuit that sends everything to the rest of the body. So this is a double circuit. And it, not 100%, but it more or less keeps oxygenated and deoxygenated blood separate. So that's a huge advance on that fish system, that single one-way fish system. But what about things like the reptiles? Well, the big thing that happens with the reptiles is they lose this skin. They lose the ability to breathe through the skin. They're dependent entirely on the lungs. And the same thing applies to all of the rest of the vertebrates. We are unable to breathe through our skin. So this cutaneous circuit is lost. Instead, this double circuitry goes entirely through the lung for oxygenation and for getting rid of carbon dioxide. Here it is here. Now, this is a mammal heart, a circulatory system rather. It's double circulation. And the two circulatory systems, this circulatory system here and this circulatory system here, are kept entirely separate from one another. In other words, you never mix oxygenated and deoxygenated blood. Never. Unless something has gone badly wrong and you've got incomplete septa or something like that. The two circulation systems operate entirely separately from one another. First of all, we receive deoxygenated blood and pump it out to the lungs where it is oxygenated. Oxygenated blood is received back by the heart and is pumped out to the body, completely separate, oxygenated, deoxygenated. In fact, this is the right-hand side of the heart. This is the left-hand side of the heart. The right-hand side of the heart deals with deoxygenated blood. The right-hand side of the heart deals with oxygenated blood and is responsible for that circulation. So this, sorry, here I am. Deoxygenated, oxygenated, okay? So we're going to see now in a little bit more detail how this kind of circulation works. Let's have a look first. This is our human heart. And what I want you to do, you have these printed as notes or print this up as a diagram because I want you to, I've tried to put this list here in an order where you can easily take a line and label the different parts of the heart. This is what it looks like from the exterior. This is our own heart, this the human heart, and we're just going to learn some of the different parts as they appear externally. First of all, these are the atria here. The atria are thin-walled, but still muscular. They can still contract and pump, but they're relatively thin-walled. Here they are, one there and one there. So those are the atria. This is the aorta there. These vessels, these great blood vessels here, these vessels are referred to as the great vessels of the heart. And one of them is the aorta, and it's here. The aorta is a huge vessel that carries the blood, oxygenated blood, to the body from the heart. The pulmonary veins, now the pulmonary, it's a vein, so it's coming from the lung to the heart. And here they are, here, there's two there. And the others, there is another pair here, but you can't stay behind the atrium here. But here's a little stublet there. So those are the pulmonary veins. The pulmonary artery is the artery which goes from the heart to the lungs. And here it is here, there. Here it is emerging, then dividing to go to each of the lungs. That's the pulmonary artery. Artery because it's leaving the heart. The superior and inferior vena cava are the vessels which take blood from the body and dump it into the heart. And this is them here. There's just, that's the inferior there behind. It's coming up here behind the heart. Here's the superior here. And they are emptying into the right atrium, and that's there. So those are superior and inferior vena cava. The ventricles are here. In fact, this is the left ventricle there, that side there. It's hard to see the division, but this is the right ventricle here on this side here. The left ventricle is much larger and is much more muscular. And the left ventricle, that is because the left ventricle pumps blood out of the aorta to the body. The last thing to label here and may take a note of, the heart itself is active the whole time, of course. It's a very active muscle. It needs a constant source of oxygen and of nutrients to keep it beating. So it has its own circulatory system, and the major vessels are here. You can see them here. They lie, the arteries and veins lie close to one another. So these here are the coronary vessels here. They're both coronary arteries and coronary veins, but you can't distinguish them. So these are the coronary vessels there. They actually emerge from this aorta. They loop over and behind to supply the heart with blood. Okay, so make sure that you have that clear in your mind, because it helps to have that idea of what the heart looks like and of its different parts when we consider the interior of the heart, the structure of the interior of the heart, and the circulation through the heart. So we're going to follow blood now from deoxygenated blood. We're going to see it coming from the rest of the body. It's been used, it's diffused all its oxygen, and it's diffused all of its nutrients, etc. But it may well be full of nutrients, because don't forget that the nutrients from the gut enter the liver through the portal system. Then from the liver, they enter the circulatory system. So although it's deoxygenated, it may be rich in nutrients and still has to be circulated. So the blood coming from the body enters the heart via the vena cava. Here's one here. That's the vena cava. This is why I wanted you to label the previous diagram. This is vena cava inferior. Here's the superior here. And they empty into the right atrium. And that's it there. The right atrium is a muscular chamber, but it's relatively thin-walled. It pumps its blood, all the blood that is in it gets pumped into this right ventricle here. And the right ventricle is a powerful chamber, has a thick muscular wall. When it contracts, it pumps the blood out and through these here, which are this is the pulmonary artery, and it divides into the right and the left pulmonary artery there, and that is going to the lungs. So it's an artery because it's carrying blood away from the heart, but it's carrying deoxygenated blood away to the lungs so that it can dump its carbon dioxide and pick up oxygen. So it is oxygenated, and that blood is returned to the heart through the pulmonary veins. Here they are here. There's the right there and the left here. These here, by the way, come around the back here to empty into this atrium here, which is the left atrium. The left atrium, when it contracts, it pumps all its blood, all the blood in it, into the left ventricle. And that is this here. The left ventricle is the most powerful chamber of the heart. When it contracts, it produces actually a high pressure in the blood. It produces a high blood pressure. And that blood is pumped out through this great vessel here called the aorta. The aorta is responsible for distributing oxygenated blood to all of the body. So it is a huge vessel. Here are important divisions of it here. You can see there, but it's curling back here, and we'll see later that it curls down to go to the trunk and supply all of the organs of the trunk. There's just one thing I want to point out to you, and that is that between these chambers and controlling the flow of blood through the great vessels, there are a series of valves. And these valves have to be able to resist high pressure, especially this side here. They're very powerful valves. These valves here are, but this is between an atrium and the ventricle. So it's called an Atrioventricular Valve. And this is one here. And it has flaps which point in this direction here. So you can imagine when this atrium contracts, these flaps will open and allow blood to flow this direction. But when this ventricle contracts, these flaps close. They are constrained. They are held by sets of connective tissue here. But they can flap closed like that. And under pressure, that is what they do. And they prevent the backflow of blood into the atrium when this ventricle contracts. Up here, leading into this great vessel here, which is the pulmonary artery, there is another kind of valve. It's called a semi-lunar valve. And it's a valve produced from the walls of the vessel. And again, it will allow the flow that direction. But when any pressure is put on this vessel here, those valves will close to prevent a backflow of blood. So the blood is pumped by the atrium and the ventricle, and its direction is determined by the valves that are present. We're going to see a very similar system on this side here. Okay, so for the moment, that's it for today. And then I'll see you again on Wednesday. Friday is your test, and then you have a break. Okay, so I'll see you then on Wednesday.